Hi, so yes, I'm Brian. I'm a PhD student in the HIDALL lab, and today, as very kindly introduced, I will be talking about neural distributions in the human brain and functional state networks. So, can we get functional connectivity from structural connectivity? The human brain is organized into correlated and anti-correlated networks, and this has been shown in previous fMRI studies. However, prior attempts by other groups to get these functional networks from structural connections have failed. So specifically, they used DTI-measured long-range associations, and they were unable to recreate these fMRI functional connections. And so our idea was to use DTI and its resulting neural fiber compositions to try and relate the distributions of anti-correlated and correlated networks found by structural connections and try to relate that back to the functional. And so first off, we have to define neuropil density in the context of this work. So when we say neuropil density, we mean the neural and glial cell bodies as well as the synapses. So these are split into the cellular density and the synaptic density. And so cellular density is the number of neurons in glia, which we can find ex vivo via staining. So for example, the big brain. And synaptic density refers to the number of axons and dendrites, as well as the glial filopodia. We measure this in vivo using PET. So I'm sure we're all familiar with the big brain and how it was collected. It's based on Merker staining, which marks the neuron and astrocyte cell bodies. And to go from this cellular density, to get the glial and neural density that we're looking for, we use this assumed ratio of 3:1, so 3 glia cells to 1 neuron in the cortex, and also the total number of cells in gray and white matter, and we take these from Azevedo and Yu. And so for our synaptic density, we use the PET tracer 11C UCBJ, which targets the synaptic vesicle glycoprotein 2A or SV2A, which is present in all vertebrates, and its staining is independent of synaptic activity, which makes it a perfect target for what we're trying to do. And this gives us a relative estimate of the synaptic density. To get a quantitative number, we again use an assumption of number of synapses per neuron, in this case around 14,000. And so, to give a quick overview of how we create these or how we get to our final destination, we first start with the MRI, from which we get cellular and synaptic density. And so each of these cells have to go through multiple processes that require energy, right? So, for example, housekeeping, maintaining the resting cell potential, signaling through action potential, as well as synaptic transmission and neurotransmitter recycling. And so those last three that I just mentioned, the action potential, synaptic transmission, and recycling, those all scale with activity, right? So as your activity increases in the cell, all these energetic contributions will also increase. And things like housekeeping, resting potential, and action potential, these scale with cell density. So the more cells you have, the greater contribution that those will have to the total energy budget. Whereas synaptic transmission and neurotransmitter recycling, those scale with the synaptic density. And so if you combine all of these together, we can create a model of the energy usage in the brain and create this energy map where we're predicting the CMR glucose in the brain. And if possible, we can compare this with maps that we measure in vivo and see if our prediction matches with what we actually observe. And from this, we can generate our energy budget. And so in silico modeling of active biological systems has increased in recent years due to improved technology and increased computing power. And it's now used in fields like physiology, cancer, anything omics related. And so the idea is to use these models to model the brain's energy usage and create these energy budget maps that we just discussed. And so the traditional energy budget maps are created using very generalized neural counts, right? So instead of having individual patient synaptic densities or cellular densities, we have a generalized idea of what it should be, and we use that for everybody. But obviously this is not ideal, and we'd like to get individualized values. And so based off of work that my colleague Adil Akif presented here last year and has published, we can use MP-RAGE and ADC and FA together to predict cell and synaptic density for individuals. And so kind of a quick overview of how he does it. So we start with MP-RAGE at a voxel, ADC at a voxel, and FA as a voxel. And then because these are only voxel-wide, they do not have neighboring information, right? So we also input some smooth versions of these inputs to the model to get some neighborhood information and hopefully make the predictions a little bit more robust. And we have a very shallow network with 40 nodes, 20 nodes, 20 nodes. And at the end, we get our SINDEN and CELDEN predictions. So we have 64 total subjects taken from the ADNI database. So we limited our subjects to only the ones that were scanned on Siemens 3T scanners using ADNI 3. And we used 10 of these subjects to train the model, and we have predictions for 54 of them. And we use BigBrain and our PET data as our standards for CELDEN and SINDEN. And so on this slide, you can see very quickly the individualized predictions that we get. So it's very obvious for the CELDEN that there are variations among individual subjects. There are as well for SINDEN, but it's a little less obvious. And so after we get our individual predictions, we can create these CMR glucose-derived maps. So first we split everything into CELDEN and SINDEN because they have different energetic contributions, as well as gray matter and white matter, because again, there are different energy usage and different energy levels in these types of matter. And then using the known formulas for how much energy we expect to be used, we can create these predicted maps. So we have one for CELDEN and one for SINDEN and gray matter and white matter. And then we compare that with this one down here, which is what we measure in patient. And so from that, so that was all work that was done previously by my colleague. And to expand upon that, now we look at correlation networks in these subjects. So using the subject-specific CELDEN and SINDEN that we previously predicted, we now estimate subject-specific CMR glucose. We subject each of these brains into the 41 different Brodmann areas using the MNI template, and then we create Pearson correlation matrices across subjects. And so after creating the matrix, we reshuffle it such that all the positive correlations are sorted or reclustered along the diagonal, just for ease of visualization. And the final order of Brodmann areas is consistent such that every metric that we look at uses the same order of Brodmann areas. So to kind of explain the reordering a little bit in visual style, so if you see in that matrix over there, we have what Brodmann areas 1 through 41, except row and column 1 actually correspond to Brodmann area 22, or the Wernicke's area. The second row and column correspond to area 37, the fusiform gyrus, and so on and so forth. It's not 1 through 41, it's this list here. And so qualitatively, looking at our results, we get these 13 Neuropil-derived networks, 11 of which along the diagonal are positively correlated, and we have 2 negatively correlated ones, or anti-correlated. And to kind of explain where we get these, so first we look at the CELDEN, right, and we compare the left brain and the right brain. And from this, qualitatively, we look and we see we have these 13 networks. And then we performed a t-test on each of these regions to ensure that they are significant. And we repeat the same thing for SINDEN and CMR glucose using those same initial networks that we find in CELDEN. And as you can see, specifically in CELDEN and CMR glucose, this one area drops out because it's no longer significant. But the other regions are conserved. And then so we do the same thing, except now we're comparing CELDEN versus SINDEN, CELDEN versus CMR glucose, and SINDEN versus CMR glucose in the right brain as well as in the left brain. So in these two slides, you can see that while most of the networks that we see here are conserved, some more of them are dropping out when we compare across modality, or not across, but across the different types of tissue. And so these are regions that have been previously described in functional MRI. So things like the default mode network, the language network, the sensory motor network, these are their locations, and as you can see, some of these networks are the exact same as the ones that we find, specifically the anti-correlated network 13, which is very large, as well as those down the bottom diagonal there. And so to give a brief summary, we identified these 13 regions, 11 of which are positively correlated and 2 of which are anti-correlated. Networks 1 and 2 have been described in previous PET studies. So in these studies, they do something very similar to us, because using PET data, and we're finding the exact same networks. And these compose of the visual cortex, the somatosensory cortex, and the motor cortex. So again, all these networks for Selden are significant. That's because that was our original template that we looked at. And network 9 drops out for Syndan and CMR glucose, as well as a few other networks drop out when we look across modalities. So there are a few limitations of this study, of course. We only had 54 data sets. We want to significantly increase this number of subjects. So if we expand past just ADNI3, if we expand past just Siemens scanners, we can get around 450 subjects, and we're in the process currently of processing those and getting those ready. We'd also like to use a stronger network for prediction. So instead of using this MATLAB feed-forward net, we would like to use a convolutional neural network, and that's been written. We're currently in the process of testing that. We'd also like to compare with individual resting state fMRI and CMR glucose PET data. So that data is also available through ADNI. We hadn't looked at it before, but we're also in the process of going through that. And hopefully that would allow us to track changes across gender, age, health versus disease, things like this. And so in conclusion, for the first time, this individual Neuropil data, which we predicted previously, has been used to show correlated and anti-correlated networks. And these networks compose of things like the limbic system, the sensory systems, and some also previously described functional networks. So these findings are very interesting in the context of previous studies that report that structural networks do not predict functional networks. We're showing that potentially we might be able to do that. And finally, the intermediary of metabolic networks traded by the energy budget demonstrates the promise of individualized metabolic data sets so that we can gain insight into the underlying mechanisms in health and disease for individualized subjects. And yeah, so a bit of acknowledgement. I'd really like to thank Adil Akif, who really did a lot of this initial work and who has helped guiding me a lot, as well as members of the Hyder Labs, Dr. Peter Herman and Dr. Famid Hyder, as well as John Rofri. Yeah, thank you.